Great, hello. Can you all hear me at the back? Yes, you're nodding. I'm Andrew Beggs. Thank you for inviting me. I've not been to one of these conferences before, but I've heard all about them. As you heard from Dilly, she is my patient. I work at my employment contract is with the University of Birmingham, but I work in so many other places. I work at University Hospitals Birmingham, that some of you may know. It looks like a TARDIS or a spaceship that's landed in Edgbaston. I'll show a photo at the end. I also am part of this thing called the Rare Disease Collaborative Network, which is a new thing done by the NHS to improve the care of patients with hereditary bowel and other cancers. So my first slide, yes, I am a surgeon, if you've ever seen it, which I know is unusual. I am also a molecular biologist, which is even more unusual. I usually get funders when I go for grant funding go, what, eh? What's that all about? But there's a gentleman who some of you may have heard of called Ian Tomlinson. You can blame him for giving me the bug for genetics, because I did a PhD in his lab, initially at Cancer Research UK in London, and then when he moved to Oxford, he told me, if he's watching Ian, I'm sorry I'm telling this story, but he told me about, he got his postdoc to tell me we were moving to Oxford, so one day I packed everything up and we moved to Oxford, and I commuted up and down the M40 for 2 years to finish my PhD. But I also run the Lynch syndrome service in the West Midlands, and I'm lead of it for what's called the South and Central region. I'll show you a map. And I also I'm also one of the centres for the polyposis syndromes, which are obviously less relevant here. So here's a map of what we cover in the purple. I always like showing this map when I go to give talks in London, because they think London is the end of the world and nothing happens outside the M25. And I like to point out to them that the South and Central, which is the purple bit, is actually quite a big geographical region. And actually, if you look at the databases, and Sir John will know this, if you look at the databases for Lynch syndrome, Birmingham has one of the largest in the UK, because we have a lot of Lynch syndrome patients. And when you add in Oxford and Wessex, it's even larger. So now things are changing for Lynch. We've made a geographical centre for services, and I do run the services for the Oxford, South Central, so Wessex, the West Midlands, and Oxfordshire. And we are beginning to make them better. It's definitely a work in progress. But at least within the West Midlands, as some people may know, hopefully they've noticed a change in the service over the last year or so. We have a new service launching. Thanks to the funding from NHS England that's now coming, we will have, by the time I finish training them, 8 Lynch syndrome trained CNSs. We'll also have a hotline number for people to call. I call it 0121 call Lynch, but they didn't let me put that on the leaflet. We also have dedicated endoscopists with GAs if needed, so I'm offering Lynch patients general anaesthetics on my list if they want them. You can't all come for general anaesthetic. You have to sort of have a go at normal endoscopy first. Having had a colonoscopy and OGD myself, though, I do appreciate it's unpleasant, especially the bowel prep. I'm afraid there's no magic way to clear your colon out. If there was, I would have definitely taken it. We also have dedicated Lynch syndrome surgeons. That's me and one of my colleagues, Shazad Ashraf. He's broken his hand, though, so he's not doing that at the moment. And we also, within the West Midlands, have a nominated geneticist, Kai Renong, who some of you may have met. We have gynecologist, Janos Balega. We have a dedicated dermatologist, because skin cancer has always been a problem, has been missed a lot. We also have a dedicated set of urologists, and we're quite aggressive with urology, even though there's no evidence for it at all. We're extremely aggressive with urology screening. We do cystoscopy, we do MRI of the renal tracts. And we have an oncologist, who some of you may have met, the inestimable Gary Middleton, who is a very tiny, hyperactive Cockney man. He's even more hyperactive than me. But he is also a world expert in immunotherapy and various different cancers. And him and I work together very closely, and we also live near each other, bizarrely, completely by accident, down the road in Leamington Spa. So, I'm going to give you the good news talk and why things are better from the perspective of a surgeon. And this is I'm going to show you some slides of a real patient, images, he's not in this audience. He's recovering at the moment. But he doesn't mind me showing them, he's given permission. But apologies if any of them are a little bit close to the bone. If they are, I'll skip through them. But hopefully I'll illustrate good news. So this is a man who presented to us in Birmingham. He's 38. Now this is an MRI scan, and I'm not expecting you to give me a detailed view of it, but you can see with the yellow lines, that is a big cancer. It's about this big. And he turned up in clinic with a cancer the size of a beach ball in his pelvis. No symptoms at all. During lockdown this was, when everybody was coming out. 38, young children. And the reason he came to see us, rather than his local hospital, is we run something called one of the pelvic exenteration services. It's a really unpleasant operation where you basically take out everything in the pelvis, sometimes including the spine. And we run one of the national centres for that. And this gentleman's tumour was abutting his spine, it was invading his bladder, it was invading his prostate, and it was invading the base of his penis as well, actually. So he would have had to have literally everything taken out. But you'll hear more about this from Kai Keane later, so that gives you a clue. This is him 3 months later. It's gone. No surgery. And we'll talk about that in a minute. What did we do? Have a think. I'll give this same talk to the medical students. What did we do? What changed? So for those who don't know, the white bit is his bladder, that's his spine, and that's his normal bowel there, and that's his prostate. It's gone. Now, I have to say, this isn't typical of all Lynch syndrome patients, but this is an example of how dramatic it can be. So let's have a quick, I know you've all had a very good chat from Ian about bowels and bowel cancer, but we know that bowel cancer is caused by a series of mutations happening. And you get a bowel cancer in Lynch syndrome, Lynch syndrome doesn't cause the bowel cancer necessarily. It's a mutation in APC, as Ian has talked about. But what Lynch syndrome does is it puts the foot on the accelerator, and it makes them develop very, very quickly. That's why all of you have colonoscopies regularly. You should be. Even at Birmingham, where we've not been great in colonoscoping people recently for various reasons, Lynch syndrome accelerates this process, and that's because of mismatch repair deficiency we've talked about. So let's talk about some more prosaic examples of what can go on. This is another example of a real patient. This is a lady who came to my clinic, 60-year-old. She had a strange family history in that she seemed to have bowel cancers on both, young onset bowel cancers on both sides of the families. And she had one polyp found at colonoscopy. Up to this point, you know, nothing, we don't know what's going on. It's very unusual. Now the polyp had a tiny focus of cancer within it, like literally a couple of millimeters. And up until very recently, thanks to the work of some of the people in this audience, they wouldn't have had mismatch repair immunohistochemistry on a polyp of that size with that cancer, but they did. And she was mismatch repair immunohistochemistry positive. And she had loss of MSH6. She had a testing, and she was found to have a mutation in the gene. Now, one of the things our new Lynch syndrome service, sounds like I'm advertising it, one of the things our Lynch syndrome service is trying to do is trying to stop surgeons from being surgeons. Because if you've, most of you in the audience will probably have met surgeons, and they tend to be quite, and I can say this, they tend to be quite binary in their mindset. Operate, don't operate. And so I had to, this poor lady thankfully came to see me. The surgeon who she was under wanted to take everything out. Let's do a subtotal click. Louder. The surgeon wanted to take everything out, her entire colon out. And I'm sure there's probably people in the audience who've had this. And you'll all agree it's an unpleasant operation. You have unpleasant side effects. You have unpleasant amount. You can get diarrhea. And it's associated with a higher leak rate in the bowel, which is problematic. So do they need a sigmoid colectomy? The evidence suggests probably not. She's 60. She's got Lynch syndrome. She's only developed a very early cancer. And we now know from studies by people like Kevin Monahan, there is actually no difference in survival between people who have a limited bowel resection, we take out just the part of the bowel with the cancer in it, rather than taking out the whole colon. There is an increased chance it will come back, but that's a time function. So if you're 60 and you have a Lynch syndrome polyp cancer, it's probably better you have a sigmoid colectomy, but you should have a discussion with your surgeon. And that's why we have expert Lynch syndrome surgeons, because all surgeons like doing subtotal colectomy because it's a fun operation. But it's not the right operation for a patient necessarily, because it causes increased leak rate, diarrhea and problems. So we now have a discussion with patients and say, well, what do you want to do? Let's give you another example. Ian touched on this before. Lynch syndrome causes a failure in DNA mismatch repair. And this causes lots of mutations throughout the genome. If you imagine a normal bowel cancer has, if you looked at the entire genome of bowel cancer, it has 10 mutations. A Lynch syndrome cancer will have 100 or 200 or 300, a lot more. And these cause spelling errors and cause frame shift. And what this means is the spelling errors get turned into proteins with spelling errors. And that's detected as abnormal by the immune system. It's called neoantigens. And these tumours are very hot to the immune system. They can be seen, but the immune system doesn't destroy them. We know now why, as you'll see in a minute. Now, thanks to the work of John, I dare not talk about CAP in the presence of John Byrne, but you know, because I'll probably get something wrong. But we know that aspirin should be and is the standard of care for all patients. It's in the NICE guidelines, and we, at least in Birmingham, try to make sure all of our patients are on aspirin. I have a special template letter for the GPs that email me and say, why is my patient on 600 milligrams of aspirin? They're only 30 years old. So they then get a 10-page letter from me with all the papers and references. But we know that aspirin is important, and it's important that all patients get it. So that's one way we reduce the inflammatory burden. That's one way we make them better. But the study that really transformed, for me and for Gary, the way we treat Lynch syndrome patients is by a lady called Miriam Chalabi, and she's from the Netherlands. She's an oncologist. She's recently become a consultant. She recently defended her PhD thesis, and she's carried out a study called Niche. Now, I'm sure Kai Keane will go into more details about Niche, and I will. But what it did show is that people who have what are called DMMR tumours, deficient mismatch repair, like people with Lynch, have a real tumour regression. So on this diagram here, this is people in the red are people with Lynch or Lynch-like tumours, and people in the green are people with more standard tumours, what's called PMMR, proficient mismatch repair. And on this axis here, this is response. So this is no shrinkage in the tumour, and this is the tumour that's disappeared entirely. And unsurprisingly, you can see that most of the people with deficient mismatch repair had a complete response to the cancer, to immunotherapy. But not all of them did, which is important to recognise as well. You can see here, this is the table. These are Miriam's slides, not mine, because I work with Miriam now. This is the effect of immunotherapy. And this table here on the left, your left, is the deficient mismatch repair, the Lynch group. And this is the percentage of cancer that was left after immunotherapy. Okay? Now, niche is expanding. There'll be more patients going into it. But as you can see, there wasn't much tumour left. But one of the important things to bear in mind before everybody gets too excited and says, I want immunotherapy forever, some of the tumours that are left were still quite advanced. So this is the T3 stage, quite advanced. So we need to be careful about how we use this new tool. Interestingly here, some people did have a response in the what's called the mismatch repair proficient group. So those who had immunotherapy but shouldn't have had a response. Some people actually did. But let's go back to our man. So what happened here? Once again, he was in our MDT. We don't have a separate Lynch MDT. All Lynch syndrome patients get discussed in our main cancer MDT, and I'm always there, or one of my colleagues. And so we had a good old discussion, which lasted about 20 minutes, and we said, well, he's got two options we can give him, because it's all about him. It's not about us, but we can give him options. You can have surgery with everything removed and all the side effects, or we can try immunotherapy. So we tried immunotherapy, and we gave him something called nivolumab and ipilimumab, which are two different immunotherapy drugs. And as you saw from that picture, he went from that beach ball to that 3 months later. With the only side effect he got was a runny nose and a bit of diarrhea. Now, once again, immunotherapy isn't that great for everybody. Some people have really bad side effects from immunotherapy. Ian mentioned CMMRD, which is a very rare form of Lynch syndrome where you have two copies. And I have a patient with CMMRD who had immunotherapy. I think she was on cancer number 5 or 6, and she got very severe, she doesn't mind me telling you this, she got very severe pneumonitis, so inflammation of the lungs. And that left her in hospital for 3 weeks. So that is it's not a catch-all sensible treatment for everybody. We still don't understand who's going to benefit from it. But what about the mismatch repair proficient patients? Because, you know, actually, interestingly, if you blast a Lynch syndrome cancer with immunotherapy, what's left, interestingly, doesn't look like a normal Lynch syndrome tumour. So one of my PhD students a while ago, Neeraj Lal, who's now one of my clinical lecturers, tried to work out why some tumours that shouldn't respond to immunotherapy do, called the PMMR set. I'm going to show you a pretty picture now. I'm not expecting you to take notes or write an essay on it. Don't worry, I said this to the students. So this is what's called this is a 2D hierarchical cluster plot, and this is a plot of patients with bowel cancer, and each dot represents an immune gene. And on the y-axis here, these are patients. Sorry, x-axis here, these are on the y-axis, these are genes. And basically, what we're trying to do is understand why some tumours are hot and why some tumours are cold. So we got a computer to look at it, and we sorted it all out. And this includes the mismatch repair proficient groups. And I said to the computer, can we pull out groups of patients with hot and cold cancers? And it pulled out 4 groups. 4 groups, interesting. Now, unsurprisingly, this group here is the hot group, and that's the Lynch syndrome tumours. And this group here is the cold group. You can see it's blue, because blue is cold, yellow is hot. I know it makes no sense, but it makes sense to people who do computers. But interestingly, in the middle, there are 2 groups here, which are warmish and quite hot, or as we called it in the thing. This is the very, very hot. This is the surface of the sun tumours, and this is sort of a long day in the desert, and this is Siberian tumours. And these patients might be suitable for immunotherapy. And these people, interestingly, this group here are the people that have tumour left after immunotherapy in Lynch, which I've put here. So clearly there is a role for immunotherapy in what's left. But one of the problems is if you've got Lynch and your tumour's shrunk right down and what you've got left isn't hot, how do you make it hot? Also, some patients with Lynch syndrome don't have a good response. So although you saw the niche trial there, it all looks very impressive. Niche was a selected group. So Miriam took patients she thought would respond to immunotherapy and gave it to them. So it wasn't randomised. So the problem we have now is we're trying to do a randomised controlled trial of this called Foxtrot 5. And the issue is, because they had such good results, none of the drug companies will fund it. Because they say, oh, niche is fantastic, you need to give our drug to everybody. But what the tumours that they don't have a good response for, the Lynch patient, we think probably about 60 to 70% of Lynch patients will have a good response to this drug. Those who don't have a response, those who have the cold tumour, can we make it hot? Now, unfortunately, I have signed a confidentiality agreement with the drug company who invented nivolumab, so I can't tell you exactly, but the spoiler is yes, we can make it hot. And I will publish it, and the company will make their fortune off me. But we do think we can make cold tumours hot now through a mechanism that I will go into detail with when I'm allowed to talk about it, perhaps at another meeting in a year or two's time. Now, Birmingham is also, apart from being known for canals, we've got more canals than Venice here, I'd like to say, and the Balti. We also are known for something called the Foxtrot trial, which is a trial. People with bowel cancer, up until very recently, didn't have chemotherapy until after their cancer was out. But Birmingham is famous for something called the Foxtrot trial, where we've given people chemotherapy before they have their bowel cancer taken out. And this allows people to have a better survival. And actually, interestingly, they had better long-term outcomes. And the Foxtrot trials just recruited, and we had about 1,000 patients in this. But what it did show, and this was completely unexpected from Foxtrot, well, it wasn't completely unexpected, it was quite unexpected, is that you had a Lynch-type tumour, whether you actually had Lynch syndrome or you had DMMR as a consequence of just losing the tumour, you didn't have any response to chemotherapy. In fact, you did worse. So we now know, that is one of the reasons why people with Lynch syndrome should all get MMR done on their tumour block before they have their operation, before they have any chemo. So that allowed us to work out what was going on. And you can see here, on the graphs here, these are survival curves of people. So these are people who didn't have defective mismatch repair, and there is a separation. That means they did better with the Foxtrot chemo. But people who had the Lynch-style tumours did the same. So there's probably a number of people in this audience who've had chemotherapy, and you probably won't want chemotherapy if you could avoid it. So if you have a DMMR tumour now, we won't be, at least in Birmingham, we don't give you Folfox chemotherapy if you're DMMR because it's a waste of time. But we might give you IO. So the Foxtrot trials are coming. They've been funded. There's also some science around it, which we've called Foxtrot Tango. I dreamt that up one day. Very original. But Foxtrot 5 is the IO groups, the immuno-oncology group, where we're going to give immune agents. It's in competition with a lot of other trials, and Kai Keane will talk to you about the Checkmate trial. But one of the sub-arms of Foxtrot we want to do is specifically study this in Lynch syndrome patients, because you are a group where this could be of the most benefit. But we need to do it in a proper trial framework, a randomized controlled trial, before we do it. But one of the things I want to do as part of Foxtrot is, instead of giving it afterwards, or if somebody's inoperable, we want to give people, should all Lynch syndrome patients have immunotherapy before their operation, if they need one, for the reasons you've seen with this man? So that's a question we're trying to answer. Trying to persuade the drug companies to fund it is a different matter, but we are getting there slowly. But one of the things I also do is I do research, I do science, and if you do unfortunately have a cancer that requires chemotherapy, we want to try and develop better biomarkers, not just mismatch repair, but other things. So I work quite closely with the groups in Leeds. I do their science for them. And they have shown there's another drug called panitumumab, which is a EGFR receptor monoclonal antibody. So it's a drug we give to a lot of people with colorectal cancer. That we can give it to people with metastatic cancer, but not everybody has a response. So they've shown, by using a molecule called AREG and EREG, you can actually predict who's going to respond to this drug. So that's great. And actually now, which I'll talk about in a minute, we're carrying out a trial of this in patients with cancer to try and better stratify them. But it's not just all about that. There are other biomarkers coming up. And so one of my PhD students, Regina, and myself, with Fedor, who's one of my colleagues in the university, we found another marker for response to EGFR, so cetuximab, some of you may have had. And it's called tetraspanin 6, which is a molecule that sits on the surface of cells, and it pulls in the EGF receptor inside the cell and lets it work. So there are multiple markers coming. We show that you can predict, once again, who does better when they've got tetraspanin. This was published in a journal called PNAS, so that was good for my CV. So the aerial study, which is another study, this has been funded. So if you come with cancer and you meet the criteria for it, we will now do a molecular test on your biopsy. You'll come to my laboratory in Birmingham, and we'll measure the AREG and EREG levels, and we'll make a decision about whether you have this cetuximab chemotherapy based on that. It's the first study to do that prospectively rather than retrospectively. So this will be yet another biomarker we can use to help people. And some people have very profound responses on cetuximab when we give it to them. Now, another thing I want to talk about is we run a, Delia said I'll do some science as well, so I'm going to do some science. I'm going to show you some cool science, as I put it, as one of my PhD students puts it. People with rectal cancer are relatively common, but with Lynch is slightly less common. Only about 2 or 3% of rectal cancers actually have Lynch or a Lynch-type tumour. But the treatment for rectal cancer is very different to how we treat colon cancer. We we always have given treatment before surgery for rectal cancer, and we give a combination of chemotherapy and radiotherapy in a lot of people. But we do know in people who have chemotherapy and radiotherapy, like you saw with the immunotherapy, some people have a really good response to chemoradiotherapy. The tumour disappears entirely. And these people don't necessarily need an operation. We know now from a study called TREK that, and a successor study, if you're into this, is called Star Trek, which is, you know, I didn't come up with the name. They can be watched and waited. So instead of having an operation, like the man you saw with the immunotherapy, they can just be watched. But we wanted to work out the reason why that happened. So a lot of research studies in science up to this point have been, you get the tumours out of the archive and you analyse them historically. Or you grow a cell line, which is not really a representative model of the cancer. So we've made something called organoid models, which are the latest hot thing in cancer biology. And what are organoids? Organoids are mini avatars of cancer. They're not the little blue men in space, as my wife calls it. They are little tiny models of your cancer that we take a sample of it, we grow it in 3 dimensions in the laboratory, and we use it to understand why some cancers respond to chemotherapy and why some don't, but more importantly, what the mechanisms are in those cancers. So thanks to the recently funded, it's called, you pronounce it mollusk, I was struggling for an acronym for it, we made a patient-derived organoid platform. So in other words, patients would come to my clinic or the hospital, and we would take a biopsy of their cancer prior to treatment, and we would use that as a model to understand what they were sensitive to and what they were resistant to. And the whole point of that project was to understand why some people were sensitive to chemotherapy and some people were sensitive to radiotherapy. And I've compressed 5 years of study into this, but we looked at the molecular differences in these organoid models to understand why some patients respond to treatment and some don't. So here's a pretty picture. Yes, ooh, everybody goes, ooh. So this is from my PhD student, Agatha. So this is to show that the models we make of these cancers are literally little cancers in the lab. And each one of those dots you can see in the screen is a cancer cell. And this is in 3 dimensions as well. We haven't put all the colors in it. But the thing to bear in mind with this is that these cancer organoids very faithfully follow what happens to a patient. So for instance, if you're a patient and I make one of these for you and I give it the chemotherapy that you're having, the responses we'll see in this will be the same as the responses we see in you. And the reason that's important is because we can then analyze it rather than you to see what you're resistant to. And we've done that across a number of cancers. But here's an example of a patient with a bowel cancer, just a very boring one. But you can see the reason it's interesting is the other very big difference between these and what were called cell lines is that they develop 3D structure. So you might have to squint a little bit on this screen, but you can see a little tube here. And that tube is a colon that's grown spontaneously from a little microcolon with a bit of tumour hanging off it. Because when we grow these organoid models, we stimulate them with the right growth factors to let them turn into what they've come from. Another thing we've done is this is actually a piece of liver. Well, it's not a piece of liver. We've grown a liver in the laboratory for a patient. We've taken a sample of their normal liver, and we've taken a sample of the cancer that spread to the liver, and we've put them together. And the reason we've done this is because when people have chemotherapy and it's spread to the liver, we don't really always understand why it works. Chemotherapy works in the cancer in the bowel, but not in the liver. And we now know from this model, one of the reasons is because the liver and the colon cancer, the cancer in the liver, they speak to each other, and they feed off each other's growth factors. And that's allowed us to understand new treatments for him. Now we can do other things. Now you may say, why is that green? That is a good question. It wasn't just kind of a horrible Frankenstein experiment. One of the problems when we're trying to understand how these tumours respond is we are now doing something called co-culture. So it's not just the tumour cells. We put your immune system in there. We take some blood off you and we grow your immune system and put it in. We get the fibroblast cells and we put those in as well. But the green is to understand which one of the cancer cells and are they being killed. So we have a microscope, I call it the robotic postdoc, and it's got a robotic arm, it pulls the cancer cells out of the incubator and it takes a picture of them with a drug on every 5 minutes for 24 hours, 48 hours, whatever you want. But the green allows us to see how many cancer organoids are being killed by the chemotherapy. And the way we get this in is with a virus that infects the organoids and makes them turn green. And the green fluorescent protein we've got from a fruit fly or something, I can't remember where it comes from. But the reason that's important, it allows us to track the progress. And these models this is actually a Lynch syndrome model. This is from one of my Lynch syndrome patients. And the reason that's important is because we've talked about these new drugs, why people do and don't respond to drugs. And so, but one of the things you need is a functioning immune system. And so one the link, if you my laboratory does not use mouse models or animal models. We just don't. For various reasons, ethical, political, we just don't do it. So we want to simulate the human immune system much better in a culture model, because in the past, when people study cancer, they do it in mice, but we didn't want to do that. And so this isn't original. Others have shown us how to do this, but we have developed the progress for Lynch syndrome. And this is a set of organoids from a Lynch syndrome patient. And these round blobs here, the non-blue ones, are the cancers, organoids for people with Lynch syndrome. And this blue cells here are the patient's T cells, so they're immune cells. And what we've done is we've taken the T cells out, we take a blood sample from the patient, and then we've trained it to attack the patient's own cancer. It's not very sophisticated. We just basically mash the cancer up and mix it with the T cells and then pull the T cells out and put them back in. But it's just a way of building a model to show how the immune system works in bowel cancer. And this is, unsurprisingly, 1 O is for organoid, day 1, day 2, day 3, day 4, day 5, and then although, don't ask me why the postdoc, Maria, my postdoc, did this. She decided to put day 7 as day 6, but she wrote 6. I don't know why. But as you can see here, the blue gets closer and closer, and at day 7 it's attacking, and at day 7 they look completely different. And that's because the organoids have been killed by the T cells. So we know we can manipulate T cells to attack Lynch syndrome cancers. And this also goes back to the project I told you about with Ono Pharma. This is the pictures from the molecule we have found to train the T cells to attack non-immune hot cancers. So we know it works. And we've got a model system now for doing new drugs. Now back to the original bit about chemoradiotherapy. When we did the analysis, we showed that it was due to a pathway called mTOR, which for the scientists in the audience will go, oh yeah, of course, that makes sense. But what it means is we found new agents we can give to people with bowel cancer in order to increase their response to chemoradiotherapy. So one of the drugs we know inhibits mTOR is metformin. Now metformin, I'm sure there's probably someone in the audience who's diabetic. Metformin is one of the drugs used in diabetes. There are some broader mTOR inhibitors that you can use, and there are some very specific ones that you can use in cancer as well. And I'm not going to go through in detail the graphs for you, but essentially the green line is what the cancer does normally, and the red line is what the cancer does with metformin, and this is viability again. So even by giving a patient metformin whilst they're on treatment for chemoradiotherapy, we can affect the viability of the cancer, and they have less cancer cells at the end of the study. And so that's quite easy to do, and actually most people can take a small dose of metformin without many side effects. So one of our clinical lecturers, now appointed as a consultant at Worcester, a guy called Rich Wilkin, is going to do a very simple trial where he gives people with bowel cancer who are not diabetic metformin before they have chemoradiotherapy to see if it makes a difference. Because although I showed you the home run of treatment, the immunotherapy, it's all about incremental gains in cancer treatment. No one treatment really is going to cure all cancer. But if we can have a toolbox of things in our armamentarium, we can really help. So metformin is an example. Now, I said about not dramatic, but then again, if you look at this graph, which is pretty dramatic, this is bowel cancer again. The black line is just normal, and the orange line is with this drug, daclociclib, that we've identified. And as you can see here, as you give more of it, patients have a bigger response. And actually, if you have that amount of daclociclib there, your cancer is going to be 60% of the cells will die off with it. If you didn't give it, they were just sitting there when you give it with chemoradiotherapy. So this is how we've identified new agents for clinical trials, by using these organoid models. Instead of doing a clinical trial on a patient, and you all have probably been on clinical trials, instead of giving a patient a clinical trial agent where we think it might not work for various reasons, but we're going to try it in a clinical trial, we can just give it to the organoids and see whether it gives us more information that it will work. Now, patients in Lynch syndrome don't often get rectal cancer, but when they do, interestingly, some of them do very, very well. And in fact, we have one patient who's lovely, lady, who, young, had a rectal cancer, had chemoradiation, and it completely wiped out her tumour for various reasons that we're studying at the moment. We think now another aspect of the response to rectal cancer is because if you have high mutation, if you have a hot tumour, giving people radiotherapy wipes their cancer out. So this is being examined in a study called the PRIME-RT trial that I'll talk to you about in a minute. So another study that's coming is called the TIC study, and it's being done with colleagues in Denmark, with a company in Denmark called Tecurex. And we're rolling this out in Birmingham, and it's for people who have gone beyond the standard and have unfortunately developed metastatic cancer. And the idea is to see the cellular models that you have seen, and use them to try and make a molecular test. So we take a biopsy, either via colonoscopy or via liver biopsy or a lung biopsy, and we grow, I call them mini organoids, they're called tumoroids, and then we expose them to a menu of treatment to understand what they might be sensitive to. And we're going to randomize people into standard treatment and treatment based on what we think the drug should be sensitive to. And from that, we're hoping to improve patients' quality of life, survival, chance of cure of cancer. And this is opening soon, if our R&D department ever do it. And so this is an example of how we're now using these sort of biological assays rather than sequencing in order to understand responsiveness. Now, I mentioned the PRIME-RT study, and this is Campbell Roxburgh. He's a surgeon as well. So he's another surgeon who wants to do molecular biology, and he's in Glasgow at the Beatson. And the study protocols here, he is giving patients who are having chemoradiotherapy for rectal cancer immunotherapy as well before they have an operation. And most interestingly, and I can't take any credit for persuading for this, but I'm glad he did it, he is including patients who've got Lynch syndrome in it. And the idea being, because they will be the best responders, see as a sort of an extreme. So we are going to see from this, at least in rectal cancer, whether people who have Lynch syndrome will have a good response to chemoradiotherapy with this immunotherapy we've talked about before. So that's one study that's coming. That's opened already. It's opened in Birmingham as well. There's another study that was recently funded by all people of Cancer Research UK, who've not always been massively good at funding Lynch syndrome related projects. It's been a bit variable, even though Lynch syndrome is one of the most common cancer predisposition syndromes that exist. And one of the questions, and it sort of fits in what John's doing with the MSI Plus assay, is can we detect people with Lynch syndrome earlier? And it's led by a gentleman called Isidro Cortez Siriano in Cambridge. And he's a bioinformatician, so he does all the clever stuff with computers we've talked about. But he came to see me because I have an expertise in a technique called nanopore sequencing, which you might have seen on the news. It's where you have a little USB-style stick sequencer, you plug it into your laptop, and you can sequence your genome on it. And we want to use that to see if we can detect Lynch syndrome changes circulating in the blood at a very, very early stage. And that's funded by Cancer Research UK. So it means, for any of you who come to my clinic, you will have blood taken off you, I'm sorry, regularly when you come, and you'll sign a consent form, if you want to come into the study, in order to see whether we can detect Lynch syndrome changes in your blood at a very early stage. That's just opening at the moment. We're still getting on with it. So there's a lot of exciting things happening. So just to conclude for my talk today, there's a lot of exciting work going on in cancer biology, and it's linked together by surgeons, by scientists. It's linked to clinical trials. But the question I always say to patients is always ask, could I be on a trial? There are trials out there. There will be one somewhere for you. It's very unusual for there not to be. And actually, we really want to recruit people to trials. And there are real options, as I hope I've shown you, for people with Lynch syndrome now. And I think my hypothesis is that the management of Lynch syndrome will change a lot over the next 5 to 10 years, because especially around immunotherapy, but also with things like aspirin screening, surveillance, and new molecular assays. And just give an example of how the science moves so quickly. There's a paper on the right here in the New England Journal of Medicine. Now, there's a mutation that happens in bowel cancer called KRAS mutation. About 40% of people have a mutation in KRAS. Even a Lynch syndrome, you get it. And it was one of those targets that they said was, quote, undruggable. You can't develop a drug for it. It's never going to happen, until they did it. And so they've developed a drug called soratasib, which has been developed for lung cancer. But interestingly, in this study in the New England Journal of Medicine, they also gave it to patients with bowel cancer. And some of them had dramatic responses to soratasib. And so we now know, now there are trials ongoing of soratasib in bowel cancer. Another one, the Beacon study, BRAF, another gene that's mutated in colorectal cancer. Not so much in Lynch, but in colorectal cancer. People up until this point didn't really have any options with BRAF mutated colorectal cancer. And so the Beacon study showed there was significant benefit to giving what's called triplet chemotherapy, a BRAF kinase inhibitor, a MEK inhibitor, and cetuximab together. And so we are getting to a stage now where cancer treatment really will be personalised. It won't be just throwing a random chemotherapy agent that's poisonous onto you. We'll be actually choosing the right agent for you. And then doing, if you need an operation still, doing a more limited operation rather than taking anything out in order to cause patient benefit. So hopefully I've conveyed some of the excitement I feel about it at the moment. Here's the laboratory. It's not a solo effort. There are lots of people in there doing various things. The people who have been working most on the Lynch syndrome immunotherapy is Toju, who has just left to become a registrar back in surgery, and Elaine, who's one of our we do let gynecologists in the lab occasionally, and she's our tame gynecologist, and we do have some tame oncologists, Ollie here. We also work quite closely with Gary, who some of you may have met before, and obviously I have a wide variety of funders. The final slide I'll leave you with is, I know some of you from Birmingham, but I also show the pretty picture of Birmingham wherever I go to prove that it's not a concrete jungle, it's a beautiful city, more canals than Venice. We've got a lovely brand new hospital, and I don't dare not say this in Aston University, but we have a beautiful university as well. And thank you very much for listening.